Shown here is a general summary of mechanisms of gene exchange as they occur both in microbial and macrobial populations. So the one we're most familiar with is this reciprocal gene exchange that occurs in association with obligately sexual organisms such as ourselves. So this is associated with meiosis and fertilization. It's also possible for all sorts of organisms to contribute to various mechanisms of non-reciprocal gene exchange. These can be within species and they can be between species. It is also possible for any DNA that finds its way into the cytoplasm of a eukaryotic cell to make its way into the nucleus of a eukaryotic cell and from there to integrate into the nuclear genome of a eukaryotic cell. Similarly, any DNA that finds its way into the cytoplasm of a prokaryotic cell has the potential to integrate into the genome of the prokaryotic cell. So here in eukaryotes, we have endosymbionts contributing to DNA that ultimately finds its way into the nuclear genome. We have other types of things such as phagocytized organisms that their DNA can find its way into the genome of a eukaryotic cell, first passing through the cytoplasm. And then we have various mechanisms such as transduction and transformation, also conjugation, whereby the DNA associated with various kinds of organisms, here these diversity of types of organisms is shown using different shapes, can find its way into the cytoplasm of other cells and from there integrate with some probability into the genome of the recipient cells. In the course of all of this non-reciprocal exchange, the more similar the sequence of the DNA is to DNA sequence that's already found within these cells, then the more likely that integration into these recipient cells will occur.